Hello, everyone. This is Dr. Karen's, and this will be my audio lecture for Chapter 1 for Microbiology for Non-Science Majors at Eastfield College. Just to give you a little bit of a heads up in terms of what these lectures are going to be, I'm basically going to go through the chapter PowerPoints like I would do if we were meeting face to face. I might be skipping around, and I'll let you know when I do that. Feel free to watch this lecture and have your own copy of the PowerPoints up or your book open and take notes. Okay, so this is Chapter 1. If you look at the study guide for the first exam, it will tell you what exactly you need to know for Chapter 1. There's not a whole lot. This is an overview of basically what microbiology is. So I'll make sure that we cover what we need to. That's the things that are on the study guide, and then I might talk a little bit about some other things in these notes as well. So in terms of the history of microbiology, the book begins by basically talking about a little bit about the invention of the microscope. This was very simple. And as you can see here in the notes, really this was occurring about the 19th century. Very rudimentary, very crude types of microscopes, but it did open up this world of microorganisms that scientists previously knew nothing about. So going from there, what are the different microorganisms? So we can categorize those into 6 groups. Bacteria, and that will be the main focus of this class. Archaea, which is kind of a branch off of bacteria. They're a little bit different, usually living in more extreme locations than bacteria, but in some ways can be similar. Fungi, protozoa, algae, and other small multicellular animals. Some of these will be covered in our lab. So just a brief introduction to bacteria. Archaea are classified similarly. Both of these are unicellular, meaning each organism is made up of one cell. They do not have nuclei, but they do have DNA. They're going to be a lot smaller than eukaryotes, so most of you should have taken general biology that focused on eukaryotic cells, like animal cells and plant cells. Bacterial cells are going to be a lot smaller. They reproduce asexually. We'll talk a little bit more about bacterial cell walls in another chapter, so I'm not going to go through that there. If you took Anatomy and Physiology 1, you probably looked at your own cheek cells under the microscope. So these 2 larger cells here that are taking up most of the image, those are your cheek cells. If you recall what that looked like, you might remember seeing these little teeny tiny dots all around those cheek cells. Those are bacteria. So that's to give you a sense for how much smaller these cells are compared to the cells you have probably already learned about. In terms of fungi, we're not going to focus a whole lot over fungi and protozoa, but the first 2 weeks of lab do cover these organisms, so you'll get a little bit of more information there. But some general features. So fungi, so think of molds and yeasts. These are eukaryotic, and that means they have membrane-bound organelles. That is different from bacteria. They do have cell walls. And like I said, molds and yeast fall under this category. Here are some examples. They show you a little bit about how different fungi can actually reproduce. They can either form these reproductive spores that kind of pinch off the ends of the reproductive hyphae, or they can reproduce by budding. Protozoa is another type of a microbe that will be the focus in lab this first week, or in later weeks, depending on which semester you're taking this class in. These are single-celled eukaryotes. They live in water. Some of them are free-living. Some of them need animal hosts, and we call those parasitic protozoa. Most protozoa do have the ability to move around, and different external features will allow them to do that. So they may form pseudopods, they may have cilia, they may have flagella. When we cover protozoa in lab, you'll learn a little bit more about that. Here are some images of different protozoa. Algae, we're not going to talk about at all in this class. The big thing that kind of makes them a little bit different is that they're photosynthetic. So these are one example of a microbe that can actually do photosynthesis. We're not covering any of those types of microbes in this class. All right, so let's see. All right, so in terms of getting back to this history of microbiology, so the study guide doesn't really ask you to know what's coming up next in these next few slides, but I think it's pretty interesting because it shows how scientists really used that scientific method to answer questions that have been issues at the time. And because they had just started being aware of the presence of microorganisms, they didn't know how to answer these questions. And so these are really great examples of the scientific method. So questions that were important at this time. So this is, again, probably the 19th century, maybe, no, this would be all 19th century. So, excuse me, questions that are important and that scientists were trying to answer. Is spontaneous generation of microbial life possible? That sounds kind of silly right now, but they really didn't know at the time, and so they were trying to figure this out. Another question, what causes fermentation? So fermentation is, we will find out in chapter 5. You already learned this if you took general biology, but fermentation is a way that cells can use sugar for energy. So it's a way to break down glucose, other sugars, into energy. This is important to people at this time because this is the process that produces wine and beer and other alcoholic products. Wine was a big industry at this time, and there were problems with contamination, bad wine, and they didn't know why, because they didn't really know how the wine was being made to begin with. They knew kind of what to do, but they didn't know the chemistry behind it. And so now they're going to start answering these questions and figuring out, well, what is producing the alcohol and what is making the wine go bad? Another really important question is what causes disease? This is back when people believed there was some divine intervention in terms of punishments in the form of disease. So they're going to make huge breakthroughs during this time trying to figure out what's causing disease and then how can you prevent it. So I'm going to just talk about some of these experiments that the scientists at the time did to answer these questions because they're really elegant and they really answered those questions very determinedly. So let's see, for spontaneous generation. So at this time, many people thought that living things can arise from non-living matter. Some people thought this was true for all living things. Other people thought, oh, just the larger living things that you can see, those cannot spontaneously generate, but maybe microorganisms can. And so there was a very famous scientist called Louis Pasteur. You may have heard of him. He's the father of pasteurization, which we'll talk about later in this semester. He wanted to answer, does microbial life spontaneously generate? Can you get microbes from nothing? So he developed these flasks that allowed him to hold a broth that would have nutrients for microbial cells in the broth and expose that broth to the air, but not expose that broth to any particulates or contaminants that might be in the air, like dust and things like that. Anything that would be found in the air would actually settle here. Settle in this neck of the flask. So what he did was he boiled this broth to sterilize it and let it sit indefinitely, exposed to the air. And after months, there was no evidence of microbial growth. And the way that they were looking for that was the broth, when it was sterile, was clear. You could see through it. Broth that is cloudy means that there's microbes living in it. And after months, this broth stayed clear. So he knew that, well, being exposed to the air wasn't enough to generate life. And it wasn't until he tipped this flask and allowed the broth to come in contact with whatever settled in that neck, then that's when he saw contamination and microbial growth. So there was something in the air that was already living, and those were the microbes. All right, what causes fermentation? Again, this was a big issue at the time because lots of money was lost due to bad wine. So there were kind of 4 different hypotheses that, again, Louis Pasteur designed experiments against or for to test these different hypotheses about what's doing the fermentation. What is turning the sugar in that grape juice into alcohol? All of these hypotheses are based on the observation that grape juice that is actively being fermented has microbes in it that can be seen with a microscope. So the first hypothesis is that fermentation spontaneously happens. So the test was, all right, I'm sterilize some grape juice and seal it. And if that's true, then that sealed grape juice will turn into alcohol. It did not. And it remained free of microbes. Second hypothesis, well, maybe it's the air that ferments the grape juice. Okay, well, let's use that fancy flask so we can expose our sterilized grape juice to the air, but nothing else. And again, no fermentation, and also no microbes in there either. So these 2 different tests did not prove that microbes cause fermentation, but it did prove that neither of these causes are true. All right, the third hypothesis, bacteria will do the fermentation. They will turn grape juice into alcohol. All right, so we sterilize some grape juice and we inoculate that grape juice with bacteria. Inoculate means you're going to introduce bacteria knowingly into this sterile grape juice. Seal it, let it sit. What happens? Oh, now it's cloudy. We can see by the microscope that there's bacteria in here. And fermentation of the sugar did happen, but acids were produced, which is a big no-no when you're trying to make wine. And that's what makes wine go bad. So we know that from this experiment that bacteria will ferment that grape juice into acids, spoiling the wine. We're trying to turn grape juice into alcohol. So what is doing that? Well, now this leads to our last experiment or our last hypothesis. Maybe it's yeast, another type of microbe. So we sterilize the grape juice, inoculate it with yeast, and seal it and see what happens. Yeasts will reproduce, not contaminated with any other microbe. Alcohol is produced. So now we know it's the yeast that will ferment the sugar in the grape juice into alcohol. And it's the bacteria that make that wine go bad. So now the winemakers know how to optimize their winemaking process to eliminate that contamination. And this led to some other ways of using microbes for manufacturing other things, other foods and other things. So this is just a list here of other foods that we eat today that are made because microbes are just doing what they normally do, metabolize sugar and other things, and it's the byproducts from those chemical reactions that create these foods. Other products as well. Did you know that bacteria actually produce antibiotics? We'll talk about that a little bit later in the semester, but think about that. Why would a bacteria create something that kills bacteria? Think about it. All right. The big question here, and this is on the study guide, so you need to pay attention to these next few slides. The really big question that they were trying to answer during this time is what causes disease? It's not a punishment for doing something wrong, or it's not because you're a witch. A lot of people believed those things at the time, or they just didn't know. And now they're going to answer this question, and that's going to be huge because once you know what causes disease, you can start working on trying to prevent that disease. So Louis Pasteur came up with this germ theory of disease where, okay, it's some kind of a germ, some kind of a microbe that's causing the disease. But which one is causing which disease? How can we figure that out? It's easier to fight a disease or prevent a disease if you know what you're dealing with. And the person behind this question is Robert Koch. He contributed a lot to the field of microbiology in many different ways, but the big one that we're going to talk about here is the cause of disease. So he came up with these 4 postulates. You need to know these postulates. These are used even today when somebody is trying to figure out what is causing a disease. And to be more specific, you're trying to figure out, 1, is this disease caused by a microbe like a bacteria? And if it is, which one is causing this disease? So then I can go and fight it. So in order to figure that out, you need to kind of check off each of these 4 postulates and make sure that they are true about your suspected causative agent or your suspected microbe. And then, and only then, can you say definitively that yes, this particular bacteria or this particular virus or whatever is causing this specific disease. So Koch's postulates are a systematic approach to figuring out which microbe is causing which disease. So what are these postulates? So let me give an example. I'm not going to use real bacterial names or even real diseases. I just want to give a very general example. Let's say there's some kind of, well, I'll use a respiratory infection. There's some kind of respiratory infection that you're trying to figure out what causes the infection. And you have a guess, and I'm going to say the guess is that you think that microbe A is causing this particular respiratory infection. How can you prove it? You need to go through these postulates. So the first postulate that you need to establish, and it needs to be true, is what it says here. The suspected causative agent must be found in every case of the disease and must be absent from every healthy host. So that means in this example that everybody that has that particular respiratory infection has to have microbe A in their system, and everybody who doesn't have this infection must not have microbe A in their system. If that is true, then you move to the next postulate. Your causative agent or your suspected causative agent must be isolated and grown outside the host. So in this example, that means I chose a respiratory infection as my example. So what you would do in this case is collect maybe a sputum sample from that individual and see if you can isolate microbe A and grow it in the lab. If you can, then you move to the next postulate. You need to introduce your suspected causative agent into a healthy host, and the host must get the disease. Now, you can't do this with people. That is unethical. And so that's why in the real world, scientists use model organisms like fruit flies or mice or even different cell lines as their patient. We're not going to inject people with a bacteria to see if they're going to get sick. That's just violates a lot of ethical standards. So I might use the term people or patient, but understand that we're not going to use that on people. But let's say you have some respiratory cell lines that you can grow, and you can infect them with different bacteria and see what happens. So that's what you would do. Once you isolated that microbe A, you introduce it into a healthy cell line and see if those cells get and mimic that disease. And then the last postulate, you need to be able to then isolate that microbe from the newly infected host. It's got to be there. Not only does it have to cause the disease, but it's got to be there. If all four of these postulates are true for your suspected causative agent, then you can say, oh yes, microbe A definitely causes this particular respiratory disease. But if you can't fulfill all of these postulates, then you need to come up with a new hypothesis. There could also be some limitations too. So maybe the causative agent is a virus and it's really hard to grow it in the lab. Then it doesn't necessarily mean that that's not the causative agent. It just means you're going to have to figure out a different way of proving that that's the causative agent, and you won't be able to use Koch's postulates there. And another exception to this would be if you have a genetic disease, you can't really use Koch's postulates as written, but there's a modified version that you can use for that. So you need to know these postulates, you need to know them in order, and you need to know why we use them, and they're to figure out what's causing disease. So now that we figured out what can cause disease, we can start preventing diseases. And that was the last question that was answered in this golden age of microbiology. So I'm not going to go through all of these people, but there's a list of people in your book that in the book talks about how they contributed to infection control and disease control. So for example, Semmelweis had the great idea that surgeons should wash their hands before performing surgery on people. And it really eliminated a lot of infections from surgery. So things like that. And there's a list in the book. There's a list right here. I'm not only those individuals, but all the people that the book talked about, even some of the things I was talking about earlier, all these influential scientists and how they contributed to the modern day fields of microbiology. All right, so that's where we're going to stop here in chapter 1. The chapter does go on and talk about some more modern ages of microbiology, but we're going to stop right there. That's the limit of what we need to know for this class. So go back, look through the notes, look through your book, listen along again to this lecture if you need to, and look through the study guide, and that'll tell you what you need to know.